The plasma membrane, or plasmalemma, is a complex and dynamic structure that plays a vital role in cell function. Its ultrastructure is best described by the fluid mosaic model. 1. Phospholipid bilayer. The fundamental framework is a double layer of phospholipid molecules. Each phospholipid has a hydrophilic, water-loving head and two hydrophobic, water-fearing tails. The hydrophilic heads face outward, interacting with the aqueous environments inside and outside the cell, while the hydrophobic tails are oriented inward, creating a barrier to water-soluble substances. The phospholipids aren't static. They move laterally within the bilayer, contributing to the membrane's fluidity. Different types of phospholipids exist, e.g., phosphatidylcholine, phosphatidylethanolamine, sphingomyelin, and their distribution isn't uniform across the bilayer. 2. Membrane proteins. A diverse array of proteins is embedded within or associated with the phospholipid bilayer. These proteins have various functions, including a. Integral proteins. These proteins are firmly integrated into the bilayer, often spanning its entire width. They can act as channels or transporters, facilitating the movement of specific ions or molecules across the membrane. Some integral proteins are receptors that bind to signaling molecules, initiating intracellular responses. Others function as enzymes, catalyzing reactions within or near the membrane. b. Peripheral proteins. These proteins are loosely associated with the membrane, often binding to the surface of integral proteins or the polar heads of phospholipids. They play roles in cell signaling, structural support, and other cellular processes. 3. Cholesterol. Cholesterol molecules are intercalated among the phospholipids, influencing membrane fluidity and permeability. They help regulate membrane stability, preventing excessive fluidity at higher temperatures and excessive rigidity at lower temperatures. 4. Carbohydrates. Carbohydrates are attached to lipids forming glycolipids, or proteins forming glycoproteins, on the outer surface of the membrane. These carbohydrate components collectively constitute the glycocalyx, which plays a crucial role in cell recognition, adhesion, and protection. 5. Membrane fluidity. The membrane isn't a rigid structure. It's fluid, meaning its components, phospholipids and proteins, can move laterally within the bilayer. This fluidity is vital for various membrane functions, such as cell signaling and membrane trafficking. In conclusion, the plasma membrane's ultrastructure is a sophisticated and dynamic arrangement of lipids, proteins, and carbohydrates organized to create a selectively permeable barrier that regulates cell interactions and maintains cellular homeostasis. The specific composition and organization of these components vary depending on cell type and function.